Hey everyone, welcome to another Toxic Google event. I'm Matt Bonjovi, and I'm thrilled to welcome to our stage today Dr. Christopher Mason. Dr. Mason is an associate professor of genomics, physiology, and biophysics at Weill Cornell Medicine, and the director of the World Quant Initiative for Quantitative Prediction. Chris has been a principal investigator or co-investigator of 7 NASA missions and projects, including the Twin Study, an examination of the impact that a nearly full year in space had on astronaut Scott Kelly, using his identical twin brother Mark as a control. Today, Chris is here to tell us about his new book, The Next 500 Years, Engineering Life to Reach New Worlds. In his book, Chris lays out a 500-year plan for undertaking the massively ambitious project of reengineering human genetics for life in other worlds. His book merges biotechnology, philosophy, and genetics to offer an exciting vision of the universe to come. Now, throughout Chris's talk, you might have some great questions popping into your head, and when you do, please go ahead and add them to the YouTube chat on the right. We will have time in a bit for Chris to answer some of these, so be sure to get your questions in early. But first, Chris, thank you so much for being here, and please take it away.
Wonderful, and thank you so much for hosting. It's really a pleasure and honor to be here. I wish I could be there in person, but I'm sure I'll see some of you in the months and years to come as the pandemic subsides, hopefully. So I want to walk you through, as you just heard, a summary of a book that just came out. Actually, today is the day it's officially released. And so very excited to be here. I'm going to walk you through some of the work in our lab over the past few years that's led to the sort of vision in the book, and then also lay out what I believe and hope will happen over the next 500 years. So I'll start with a little bit of background. So first, it's worth noting, as I again describe in the book, that more than ever, we are a spacefaring species. So this is the number of objects launched into space in the past few years. We're going to reach an exponential growth here in the number of objects launched into space. So really reaching a new era where a lot more things are being put up in space. Of course, as most of you probably saw just recently, we also saw that the little Perseverance helicopter that was hiding in the belly came out, just did a little launch, went up in the air, did a little quick turn, got its bearings, and then also managed to look around the landscape and even then land back safely back on the regolith in Mars. So we had the first powered flight on another planet just yesterday. Really extraordinary, exciting times. And so this is leading to, a lot of future developments, which I'll describe today, with other experiments on Mars that are coming. And so, as you can see here, sort of the vision for NASA and for other space agencies is to get to Mars using science and technology in a variety of different means to not just visit the red planet, but to eventually stay there long term. And as I highlight in the book, and I'll tell you about today, there's really this extraordinary, 2 engines of biotechnology pushing us forward towards this goal that are really going to make this possible. And they're coming mostly from a lot of work in the space sciences and cosmology, but also in genetics and biotechnology. And one of them that's really driven a lot of this is just the ability to sequence DNA and to build genetic catalogs has gotten much, much cheaper over the past 10 to 15 years. So this is a chart of what's the cost to sequence one human genome. It used to be hundreds of millions of dollars, and has recently come down to less than $1,000 to make it so that this is moving faster than Moore's law in terms of the pace of technological change. This actually represents the fastest pace of technological change in human history that's really ever been experienced. So it's really extraordinary because now we can sequence things that before we could barely imagine for obviously the human genome. So we can have at the hospital at Cornell, we call it genome-guided medical care. But we can also use it to just discover new things. So for example, we're still finding new human genes. In particular, in the past 10 years, we've added 10,000 genes total to the top, you can see here, of the number of human genes. The fundamental facets of human biology and genetics, we're still finding them due to a lot of these improved methods for sequencing that have become a lot cheaper. And so that's for the human genes. We're also using these methods to find entirely new species. So for example, this discovery in the past several centuries has continued to rapidly accelerate again at an exponential pace, except for birds. We found most of the birds so far, you can see in the middle. We're probably not going to find too many more birds. They're kind of big and easy to spot. But a lot of the other sort of families and genera of kinds of life, we continue to find more and will likely find more of them. Even in space, we recently published a paper a few weeks ago. We found new species of bacteria that had rapidly evolved on the space station, and I'll talk about these a little bit later. But what this means is basically this rapid pace of discovery. It means that every single day as a geneticist that I wake up is actually the best day to ever be a geneticist, because there's more data that exists on that day than any day prior, meaning there's greater capacity for discovery, there's greater ease of sequencing to enable more discovery, and we can better annotate and characterize all these different organisms. And this is really exciting because this led to a lot of these ideas of everything that we're discovering and then blending them into learning about the adaptations of all of Earth's life forms and how they've adapted to stress or radiation or hypersaline lakes, and use them for actually even human therapeutics. And I described this really in the book, which again, I'm excited is out today, is really my collection of dreams and hopes and actually a proposed new philosophical duty for humanity. But before I get into the future, I want to catch you up on how we got to where we are today in the past 10 years. So I put this online on our lab's website about 10 years ago. As this would be a 10-phase plan for basically the next 500 years. And the first phase was really, we would just continue to annotate the human genome and begin studying outlier humans like astronauts and begin doing a lot of work in synthetic and systems biology. And I'll walk you through some of the studies that we've done in the past 10 years that contribute to that and how that catapults us forward into the future years. So in particular, as I kind of alluded to before, we want to go to Mars, we want to go to the moon, but, NASA is not the only group that wants to do this. Also, Israel has recently had a private lander that landed on the moon. It kind of spilled out some tardigrades, or what are called water bears, onto the moon, and these can survive even in the vacuum of space. So that was kind of a, oops, you suddenly had these cute little water bears floating around the moon. They probably all are dead now, but they are technically some at least tardigrade graveyard that's there on the moon. The Chiang 5, one of the missions from China, also got plants to grow on the moon recently. So we know that if you think of cotton coming from the moon, your lunar plants could someday come to you and you could wear them. There's also plans now to build an entire moon base with the European Space Agency, and there's architectural firms already building out these plans. So if you have any kids that you're talking to later today, you can say, what do you want to be when you grow up? You can tell them a lunar architect is a legit job they could plan for, and here's a rendering from one of these architectural firms. However, to go to the moon and to actually land there and be safely and to come back to Earth, there are some things we know that are risks. So for example, these are nuclear trace recordings that were worn on the ankles of Neil Armstrong that show these high energy particles zooming through these tracer packs, which means they're also zooming through his body and causing DNA damage. So radiation is one of the big fears we have and risks that we know is present for a future spaceflight. We also know that other things change when you go just as you age. So for example, your telomeres will shrink over time. These are at the ends of your chromosomes. They slowly get chopped off and shrink over time and really are an indicator of your age. And that's at the genetic level. We also can see epigenetic changes, or this regulatory layer behind DNA. As DNA goes to RNA and proteins, the epigenome itself has these changes that can normally occur. So for example, across different bases, you have these red epigenetic marks, whereas the genetic sort of bases, the A's, C's, G's, and T's are here in black, stay the same. But you can even see a damaged DNA, like 8-oxoguanosine, is when you see basically adducts and sort of radiation can do damage. We can detect this as well. That's kind of an epigenetic indicator. But we've seen, if you look at just normal individuals, like identical twins, take them at 3 years old versus 50 years old, as they get older, they're epigenetically more distant from each other. So they accumulate mutations, so they're genetically different more as they age, but they're also epigenetically more distinct as they age as well. So this is an intriguing fact that identical twins, if you see them every given day, you see them, they're slightly more distinct from each other, both genetically and epigenetically. And we've also known that people accumulate mutations just as a consequence of aging. So one of the other things of work that's come out of Cornell is that something called clonal hematopoiesis is something we've studied a lot lately, where you actually have indeterminate potential because it's not clear what these mutations mean yet. But in more recent publications, that essentially everyone is accumulating these small mutations in their bone marrow. Eventually, they can predict whether you're going to get leukemia or cardiovascular disease even decades before you actually get it. So it can tell us, are you on the path towards getting one of these diseases, or are you sort of staying stable, much like a mole on your back? Is it staying the same size, or is it getting bigger? And this is some of the more recent work from our lab and others. So that's what changes normally, on Earth. But, what about in space? What happens to your body when you go into space? So we had a unique opportunity to look at identical twins a few years ago. This is Scott Kelly and Mark Kelly. You can see Mark always says he's the handsome one with the mustache. He's also now Senator Kelly. So we have the first senatorial genetics sampling and sequencing project to date. Basically, we had a unique opportunity because NASA said, well, if we're going to send Scott up to space for a year, we have a unique chance to do an identical twin study and do all the same measurements. So thus launched the NASA twin study, and we were one of the 10 teams selected for this mission. And we're very excited because it gave us a chance to really study almost every modality of biology we could. So, for example, when he launched up into space in 2015, we knew that he probably wouldn't be exposed to too many pathogens, but it would be good to get a sense of, well, what does vaccination look like in space? So this, for example, here is Scott Kelly giving himself his own multivalent flu vaccine, on the space station. And you can see here, just giving himself a quick vaccine, kind of making what I would consider to be the vaccine face. and made it successfully in, gave us kind of a smile with the ouch. And then also, we drew blood shortly thereafter to look at his T cell response, which we could see mimic actually a response to the flu vaccine, also his T cell repertoire changes. So this is kind of him giving us a good thumbs up. It went well. So we got frozen blood samples. We also got some fresh blood samples during this mission, as best demonstrated for phlebotomy in space by astronaut Wakata. And doing phlebotomy in space is a little bit different from doing it here on Earth. You can see here, the tube doesn't quite stay in the one plot. So even though you've got a butterfly needle, it should be pretty stable. The tube floats around, but it does fill up. There's negative pressure in the tube and there's positive pressure inside your body. So it still has no problem filling up the tube. And the astronauts are extraordinary. They don't just do this sometimes once. They'll sometimes do it 8 or 10 different tubes, EDTA or CPT tubes, you can see up on the right. So phlebotomy in space become really exciting. And we even got some of them fresh blood draws that would be dropped through the atmosphere with a chute deployed, make its way into Kazakhstan, and retro thrusters would fire. These would then be picked up by helicopter, repatriated, and brought back to Houston. And we can get cells that were freshly in orbit only 36 hours prior to also do processing for some of the T cells and immune function that we did on the study. So that was kind of some of the logistics of the mission. It was a really fun year, a lot of work to get it set up, but it was really great working in tight collaboration with NASA for all these measures. But then after a year in space, eventually Scott did have to come home. This is him undocking from the space station after 1 year in flight. You can actually see the Earth rotating down below him in the lower right corner. If you can read the Russian there, it says, "Bring back more vodka." No, it's just the telemetry data. It doesn't say that, but it does, you can see the Russian info there. The chute deployed safely, made his way back to Earth. And we were very happy, and he brought himself as well as a lot of samples that were frozen back with him, and kind of get helped up by the other cosmonauts, gave us a thumbs up, and overall was doing fine. But he did have some other changes. When he got back to the United States, he really had a severe reaction, and he felt like he was on fire. Basically, he had rashes and discoloration anywhere he had contact. He basically couldn't even have the weight of clothing on his skin. He had to basically walk around his house nude just to avoid touching anything for a few days. Some people might do that recreationally. He was doing it for medical reasons. He had flu-like symptoms. He wanted to go to the emergency room. And the big question we had is, why? Just what happened inside of him? So we know some things that happen normally is you actually get, you go about 2 inches taller because you have the lack of gravity. You get about 2 liters of fluid that moves up to your body, which definitely drives some of these changes. But this was really, severe. Nothing like this had been seen before for other astronauts. So we did a multi-omic analysis of human spaceflight. And, when you don't know where to look at two people when they're very different, one in space, one on Earth, I think the best place to look is everywhere that you possibly can. So in this case, we looked at genetic changes, epigenetic changes, RNA changes, protein changes, chromatin and antibody titers, B cell, T cells, telomeres, cytokines, metabolomics, microbiome, cognition, and vasculature. And we did this all about every 3 weeks for about 2.5 years to give us this complete profile. And what's extraordinary is when we put all these data together, we could see a number of things moving in concert with each other. So across all these different omics, which is the genome, the microbiome, the epigenome, transcriptome, all these metabolites, we could see things, moving and shifting with each other. And we published this about 1.5 years ago, is that we could see some things that were expected, like species that we knew had changed in other missions. But one thing that was really striking is that his telomeres actually got longer in space. You can see here. So this was kind of a really surprising feature. And we also saw that, maybe this was just him, but then we did 2 other astronauts and reported this about 6 months later. So it looks like we confirmed it again. And then we thought, well, these 2 showed it, but where else could we pick this up? So we then did a study with twins, 2 twins that were actually climbing Mount Everest and kept their other twins at sea level and replicated it again. That seems to be a mixture of hypoxia and stress and radiation on the body. Here too led to higher telomeres for the twins that were on Mount Everest, as well as now 11 other astronauts. And so far we've seen this seems to be a very consistent feature of spaceflight, is that your telomeres will get longer, which is a mixture of response to low dose radiation, some cellular cleaning, and even cell damage, getting rid of the cells that were closer to dying, we think. But there's other factors at play, as I alluded to for the Everest twins. So for example, in these changes, these genes that go up and down, which are these different colors here, we saw some genes activated, some genes turned off. And this gave us, interesting clues like hypercapnia. There were some genes for when you have too much carbon dioxide. So Scott Kelly actually complained about this in his book, and we thought, well, that's weird. The space station has pretty well regulated carbon dioxide and oxygen levels. And we talked to some other astronauts about what might be leading to this. They said, well, it's sometimes if you're not by all the fans on the space station, you can get little clouds of CO2 that can build up by your face. And we said, well, what do you mean? So, well, if you blow a bubble in space, for example, the bubbles don't just fly all over the place. They can even stay a little bit by your face, is best exemplified here by Jack Fisher, is you get these little mini clouds of CO2 that we think we saw here in the blood work as well. So really interesting, strange phenotypes from the unique environment of the space station. And jumping back to what happened to him when he landed back on Earth, we looked also at the immune system and cytokines. And these were some really interesting signatures that spiked up. You can see all these are different cytokines and inflammation markers that were a mixture of pro and anti-inflammatory markers that showed the body rapidly trying to deal with the stress of returning back to gravity. So in particular, some of these markers are ones you'll see there when your body's under stress or infected. Even IL-6, for example, is something we saw in COVID-19 patients over the past year. That was surprising to see because you wouldn't expect it necessarily from spaceflight, but we can see it here spike up as well. So when we think back to his ankles going up to the size of basketballs, we think these are the markers that really drove him to have that really strong immune response. And that response, plus the fact that if you look at how these genes and molecules are all connected, a lot of them deal with dealing with muscle regeneration and muscles use is what led to a lot of the really significant changes when he landed back on Earth. So looking at some of the other metrics of what happened to him in space, we wanted to look at the epigenetic age, for example. It looked like it was mostly stable. You can see here, Scott, you're in blue and Mark in green. For both of them, before launch and return, they stayed approximately close to where they began. So we don't think this was a big change, which is kind of some good news. We also had some potentially good news is that his clonal hematopoiesis, or these mutations in one gene called TET2, actually got better in space. So you can see here actually, the amount of mutations being carried in his blood for this gene actually improved, whereas other genes stayed flat. But then, when we look, what that means, maybe he got a little taller, maybe a little bit younger. Of course, the 2 inches did go away when he landed back on Earth. But we noticed that, it didn't just stay that way. So when we've been continuing to monitor both him and his brother, some of these mutations change over time, and one of them did increase later at a later time point. So we think that, again, this is very much like a mole. If you think of any mutation in you, really in any part of your body, it's not just a question of do I have a mutation, but is it getting bigger? Is it changing? And in this case, this one's going a little bit in a direction we don't like, so we're keeping an eye on that. But this is also something that's becoming more of a standard measure for other astronauts, which I'll talk about shortly. But we like to say that overall it was good news across all these different omics measures. Most of these features were plastic and recovered, and the ones that seem to be some DNA damage or cognitive changes we're still keeping an eye on, but are nothing that's kind of a red flag, I think, for Mars missions. So it's kind of a bit of the, in the past 10 years, then we've learned a lot about the ways to do these studies and to do multi-omic integration, and which gets us to, what's the next steps? This really gets us, I'm excited to say we're now entering, we're just now getting into sort of phase 2 of the plan I penned out about 10 years ago, which is to do more astronaut studies, to integrate new elements into mammalian genomes, and eventually get to Mars. So, as I alluded to, we're doing more of these studies. So there's actually 583 people that have ever been to space. But coming in September, we're working with SpaceX for a new mission, which is to do the inspiration for civilian astronauts, or what's also called commercial astronauts. So basically replicating as many of these measures as we can during some of these shorter missions into space, and also working with NASA on some other missions out to the year 2033. We were one of the other recently selected proposals to look again at clonal hematopoiesis, genetic changes, and all these other omics measures on astronauts over the course of their new long duration missions. And so that's some of the work we're doing. The other work we have worked on in the past few years is, it would be just better if we could sequence on the space station or maybe on Mars directly rather than having to bring everything back to Earth. And so, since it's NASA, we started a mission with them. They said, well, if you're doing a mission, you're going to need a patch. And so, great, we got a patch. Fantastic. The Biomolecular sequencer project. You can see this is a little, what's called an Oxford nanopore sequencer, zooming into space. And as we started planning out this mission, doing some of the other twin study work, we had a resupply rocket that unfortunately blew up on its way to the space station. And we had this really sad moment because there was nobody on board, but a lot of the supplies and essential reagents for experiments and food all went up with the rocket. And after this happened, I got a really nice letter from NASA that just said, "Dear Dr. Mason, we know that space is hard, things explode, but stay in there. Don't give up hope. We're going to get more supplies to you and get the mission back on track." And we thought, okay, this is great. Let's keep going. But in the meantime, we thought, well, let's actually try on this parabolic flight simulator, or what's called the Vomit Comet, a quick test of can we do sequencing in zero gravity, because we didn't really know at the time if we could. And so, at a very fun opportunity with Dr. Feinberg, a colleague and collaborator, we said, well, I've got this chance to go on the flight simulator. I'll try and do pipetting. And normally, if you do pipetting, if any of you have been in a lab before, normally you want your tips to stay in one place. So this is really not remotely ideal in terms of how pipetting should go, but he did manage to get it to work, even as tubes and tips were flying all around him. Eventually got back to the bottom of the parabolic flight, and everything crashed back down, maintained its composure, was going fine, loaded a flow cell, this mini-ion little sequencer flow cell. It's this big, and we showed for the first time that you can do sequencing in zero gravity. And then, fortunately, Kate Rubins was the next astronaut to be flying up, and so we got her trained to do also the sequencing experiments. She safely made her way back to the space station, and we're very excited that for the first time we got sequencing working in space on the space station. And so that means that we know it's possible, and she just got back on Earth actually 2 days ago from her follow-up mission. So really great to have Kate back on Earth. And now we actually have, this is a deployed space station certified technology. And so what's exciting about this, okay, we're measuring things. This has been wonderful. Now we can measure them even faster. We can measure them on the space station. We can measure them potentially on Mars. But I want to transition from not just measuring things, but to instead doing more active defending of the biology in our cells. So in particular, we want to measure them in general, but we want to see where they're going as well. So in particular, we are measuring things also in the clean room. This is actually where they build the rover, the Perseverance rover. I think all of you have just been tracking and heard about, and I showed you in that third slide, is this rover carried with its small helicopter. You can see when they build the rovers, though, they actually do, track essentially what microbes might be there. They look at the DNA that's left behind, but they're human. Sometimes they'll take a selfie with the robot that they're sending to another planet. I get that. I think I would too. But what's interesting about that is this has implications for what's called planetary protection, which means we have to look and trace as best we can. Is there any molecules that we're sending to Mars, or is anything that could come back to Earth that could have any biological or health implications? And so, for example, we know that there will be a Mars sample return mission. So eventually the Perseverance rover will tool around Mars. You can see these are actually some beautiful images of Mars. Find a good spot, maybe look out on the horizon. This is actually a picture from Mars. It's actually almost looks Earth-like. And it'll find a spot on the ground where it thinks would be a good place to take some samples. It'll dig into the Martian regolith and then actually, collect some samples. It might, as the evening falls, look back at Earth and see us in the sky and package up those samples. And in the year 2032, it's estimated that these samples will then come back to Earth from this Mars sample return mission. And so one of the questions we have is, well, if we're measuring and tracking and thinking about defending what could be coming, what if what's there, could be potentially dangerous? But what if it also just could be some of the DNA from these people, Jane and Joe engineers, who were just leaving human and microbial DNA on the surface? To what degree can we track that? And so one of the things that we've been building out is an infrastructure to do just that, to think about defense at the planetary scale. What's really planetary genomics. In particular, we just published work showing that there's a framework to characterize all the organisms that might be able to survive the trip and also make their way back and forth potentially to 2 planets. And then also a global K-mer index of what essentially is any piece of DNA that's ever been indexed on Earth, working with Andre Colles and Gunnar Reisch in Switzerland. And to show you quickly what that looks like, actually, is we basically have an index of every single piece of sequence that's ever been built, that's obviously in some of our databases, but also anything that's in a public database. So now we can actually search, given any sequence of DNA that's ever been seen on Earth, where has it been seen before at different similarities, and also match it relative to our other metadata. And we can also then visualize these in what are called De Bruijn graphs. So you can play with the connectivity of essentially different fragments of DNA that have ever been observed in the history, really, of any sequencing data ever. And so this is kind of a fun sort of interface that we've been building out for planetary protection, and as I was just describing here. So that is kind of what will be, I think, the end of what we'll be doing in the next 20 years as we start to get samples back and forth between these 2 planets. Okay, so that's kind of looking at the next 20 years. Hopefully by then we've begun long-term human trials in genome engineering and look at off-target effects and hopefully fix them. And we'll need to do this because we'll start to think about, can we think about other ways to look at the damage and again, be more active about our defenses. So in particular, we know that from papers we just published last year that astronauts can have this damaged DNA, this 8-oxoguanosine, this basically indicator of broken fragments of irradiated DNA showing up in the urine, like the damaged DNA is leaving their body at higher levels when they're in flight days versus before and after when they leave and return, days pre-launch or return. And we know that, that's just for missions like to space, right? Those are under the Van Allen belts in near Earth orbit, basically, or in low Earth orbit, whereas here, that's only 146 millisieverts. So it's not that much radiation that Scott Kelly had relative to some other missions, or even relative to living on the surface. Or if it's a 3-year Mars mission, it'll be much more radiation. It'll be the hardest mission yet by far. And so it leads to questions like, can we genetically protect astronauts the same way we do physically and pharmacologically? And really begs big questions like, should we do such a thing? What are the ethics and the genetics of making humans potentially fit for Mars? And we know for sure the way that this shouldn't be done. For example, He Jiankui actually, CRISPRed embryos and then released them onto the world and said, here's my CRISPRed babies. We instead are doing things in much more open and transparent methods. One project called the Genome Project Right Consortium to make all these methods and protocols and ethics very widely discussed and debated. But it's worth noting that the idea of editing cells for therapeutic purposes is already really widely deployed, especially in immunotherapy. There's a lot of CAR T trials or chimeric antigen receptor T cell engineering trials. We basically take cells from patients, modify them. You can see here, basically pull them out, modify the receptor, grow them up, and then reinfuse them back into the patient to target the cancer cells. And so this isn't just one or two clinical trials. What's extraordinary, and I talk about this at length in the book, is that there's more therapeutic editing trials than ever before. Like all these CRISPR methods of cells then being put back into patients has never been higher. So it's this extraordinary time where the idea of editing cells therapeutically is really rapidly expanding. And it really does beg that question. In the 2040s and 2050s, could we take lessons that we're taking from immunotherapies and apply other genes from other organisms? So for example, elephants, you can see how many copies of P53 they have, which can detect DNA damage. They have a lot more copies of it that they regulate quite well and have a lower risk of cancer, it seems. Or what if, for example, we take a little cue again from the tardigrades, which can survive in the vacuum of space, characterize that gene that's been discovered called DSUP, which is a DNA damage suppressor protein. It was shown that if you give them a temporary boost to this protein, they can reduce DNA damage by 40%. We've been working with this in engineering stable cells in the lab, where you can see if you put in DSUP into these cells, they can actually go right to where the double-strand breaks are occurring. And so we've been looking at this for samples with 1 gray versus 0 gray, so giving more radiation on top versus the bottom. And what's exciting is we think we can get these cells, human cells, down to about an 80% reduction in that 8-oxoguanosine. So it's dose dependent, and we actually see that this is a mechanism, basically a genome editing mechanism to protect the human cells that now is working, in our lab and other labs looking at this as well. And that's just the genome version of editing, which I think really extraordinary about the field in the past few years is if you think of the canvas of editing, you think of CRISPR as a way to basically modify the canvas of life. There are other ways to do this with epigenetic modifying enzymes as well that are small tweaks to the Cas enzymes. So in particular, this has caught the eye of DARPA, which now has an entire program that's looking at, can we preemptively express protective alleles and response elements? Can we activate, say, DNA damage repair genes before someone goes into a high radiation area or biological threat area? So this idea of preparing genes in advance and then activating them and then deactivating them as you need is also something we're deploying now in lab, and as well as DARPA and other laboratories to really think about this plasticity of genome regulation. So that I think is, be phase 3. Phase 4, it's going to get a little bit, I'll be a bit more vague as we go a bit more distant into the future. But I think all these things are based on what are known plans and also based on current science. So it doesn't require any great leap or any great invention. It's everything that we know today. So I think in the later phases, we'd start to test these protected human genomes in space environments, and we would begin to actually remove molecular ineptitude. So for example, one of them is there's a gene for vitamin C synthesis that's in our genome. It's still a pseudogene. It's still there. Why not bring it back? So, for example, if you look at other mammals, you can see the ones bold here. They can still make all their own vitamin C. If any of you have a dog or a cat, it's walking around right now making its own vitamin C. Whereas you can see a lot of primates have lost this long ago, including humans. But we still have almost all the components of this pathway for synthesis in our genome. It's just been inactivated through mutations. We could put it back. We could make it so we can make our own vitamin C, which you might want to do if you have really long-term missions ahead of you, say to Mars. So it's one kind of a relatively simple fix. But we have also, if you think about it, we have 9 essential amino acids that we need from our diet. Why is that? Other organisms do make all their own essential amino acids. And if you look in black here, the human genes and pathways, we could borrow genes from other organisms that we know can synthesize all of their amino acids. Like this is one idea that we could begin to reconstitute the entire biochemical pathways for self-sufficiency, or what's called a prototrophic human. And again, some of this work in our labs and Harris Wang and Jeff Boku that's kind of show that this is definitely possible as kind of a way to increase self-sufficiency. And the other thing we can start to do, of course, is to repair disease genes. This has been shown already for a lot of embryo work. There's actually some experiments you can see here for Shukat Metalopov is actually showing that you can repair genes for hypertrophic cardiomyopathy or other severe disease genes. And you can actually get rid of the damaged genes actually before it's too late. And there's even other modifications that have come up that have been shown that you can have even same-sex parents have their own pups. You could have 2 females have enough genetic information and egg to make their own babies. Same with two fathers having a child. There have been now 3 parent children that have already been born where you have a donor mitochondria. And what I'd say this gives you is really a complete sense of cellular liberty, from the very first cell, that every cell won't be beholden to the DNA that you got accidentally. It would be the end of accidental evolution and the beginning of guided evolution, something that would be much less, I think, subject to the slings and arrows of the universe's whims, but also would enable a kind of liberty that I think I talk about in the book is a cellular liberty, that you should be able to do anything you'd like with any of your cells. This, of course, raises big ethical questions. So if you're going to do anything you want to any cell in your body, how do you know you're going to do it right? For example, a recent discussion from Lear Patcher is like, what is the moral ability to select embryos? There's a new company called Orchid, which is starting to roll this out, where they look at both parents and help with embryo selection. And this is because it's based on something called polygenic risk scores. Is if you take a look of all the different genetic loci in your genome and these different genetic markers, can you predict what the weights are for which basically genes you should select that you give to that one embryo? And the idea behind it is you add up all these scores, you get sort of a high or low risk score of what's your high risk score or low risk score, and try and optimize to get the lowest possible genetic risk for disease. So conceptually, it's very simple, and it's just a really simple summation. But the problem with that is that the polygenic risk scores can be very biased. And so one of the great examples of this is they're biased a lot based on the ancestry. So if you have imperfect measures, you'll get imperfect metrics. So for example, if you look at the ancestry, whether you're Asian or, so look at Caucasian ancestry or British ancestry, your polygenic risk score for schizophrenia depends more on your ancestry than it does actually on your risk for schizophrenia. So clearly this is an early stage of being able to even just understand and model the genetics. And so it's clearly not really ready for prime time, but that's one reason why I put this about 50 years in the future. I think we need probably at least 2 to 3 decades worth of pure information and modeling to make these scores better. But I think it's a question of when, not if. It's a data problem, not necessarily a modeling problem, although both can be improved. And so to that end, I also proposed a bit that we also need to think about this, not just of what is your risk that you give to one embryo, but what does this look like between generations, and what's also the socioeconomic impact of gene editing, of embryo selection? What does this do to the ability of for your quality of life, or also those in your family? Can they have an improved quality of life if you have less disease? How well do we understand pleiotropy, where one gene has multiple functions, or heritability, or the severity across generations, and the economic burden? So I think all these factors need to be more built into some of these scores, essentially, which I described in one of the chapters, is how do we better understand this and have a more responsible view of not just picking embryos for one feature, but for an individual as well as a collective understanding of its impact. Okay, so assuming we get through all that and we completely understand genetics and modeling, it's getting much better. At that point, we could start to think about going towards other planets. Maybe we can do this safely and successfully, and by getting into the later part of this century, do it and start to synthesize genomes from scratch. So we know we can do this already with E. coli. We can synthesize whole genomes from scratch. And you can start to get creative at this part of our future, is that, well, if we can synthesize genomes from scratch, could we get smaller genomes? Like, could we add a whole organelle to the human biology? Like, could we make it so you could have green skin to photosynthesize, for example? Could you do that? Like, so say you wanted to go lay out by the pool and you think, oh, I'm kind of hungry, I should lay out my skin because I want to eat. Could you do this? Could we even chemically do this? So I calculated this out in lab and sent this out to lab one day and said, well, how much of your skin would be actually exposed? What's the photon loss? What's the photon capture? So making some basic assumptions about this green skin. And how many megajoules do you need to survive 1 per day? It turns out if you just want to lay out your skin for 1 hour, you need about 2 tennis courts worth of skin to pick up enough energy to last for the rest of the day. So it's not too bad. If you lay out for 2 hours, you just need 1 tennis court worth of skin. Some people might find that to be kind of a gross image. I just think it's interesting for a calculation, but that at least we know approximately what it would be in terms of the amount of skin that you would need. And if this seems kind of weird and unnerving or gross, to try and make like chimeric creatures that have both human and plant components together, it's already happening. There already have been human pig chimera embryos. There's been human monkey embryos that have happened just last week. So we know that this kind of work of blending sort of the lessons of biology from all organisms has already begun. And I think in the next century would certainly get much more accurate. I think green skin is maybe a bit of a stretch, but it would certainly be fun and be fantastic. And it would also, again, make it you more self-reliant. If you're on a planet that has very little sunlight, every bit of energy you can capture from every photon would certainly be very welcome. And again, we don't have to just look at these organisms that we know now. We also, in the phase 6, will expand out some work that we've done in the lab called the Extreme Microbiome Project, which is to get as many lessons as possible from extremophilic organisms that can survive in really harsh planets. So for example, the other planets in the farther reaches of the solar system would require a lot more, I think, genetic tools that have been found by other organisms or really evolved in other organisms and use those. So we have been looking over the past 10 years at a variety of different locations on Earth that have really peculiar, environments, either for salinity or temperature or pressure or moisture or toxicity, and using those basically to try and understand how other cells could be adapted to survive in them. So one of them actually, for example, we just recently found organisms that could that evolved essentially to be quite distinct species on the space station, had new genes for DNA repair, even biofilm formation, even molecules that we could use potentially for new sunscreen. So I think, if we use the lessons of evolution from every creature on Earth, that gives us a much larger genetic toolbox with which we can deploy our own survival when we start to go towards farther planets, not just Mars, but even to look out beyond Mars. And so my hope is that that'll start to happen around the year 2100, 2150. Now, eventually, when you look out a bit farther into the future, we would potentially start to send new genomes to seed Earth-like planets to actually start to go to Mars, not just to visit, but again, just to make it more self-reliant. And so we've published methods and protocols that really put this out over multiple stages, where eventually you'd want to have on-demand pharmaceuticals and therapies that are created on Mars and large-scale crops and fermentation methods. And so a lot of these methods we detailed in a paper last year that we know it's actually possible, and I talk about this in the book a lot, is that there are ways in which we know the chemistry exists today. It's just about getting the reclamation and recycling methods to work well on the Martian landscape, but also eventually could work on other ones as well. And what this would then give us, much like cellular liberty gives you an ability to have any cell do anything, planetary liberty gives you the ability of those cells to live on any planet. So one of the big ethical critiques of, say, modifying a human to go live on Mars is, what if they could no longer come back to Earth? If any of you have seen the movie, it's the series The Expanse, or read the books, people who grow up on the asteroid belt can't come back to Earth because the gravity is too punishing. But what I would always strive for is to not have less planetary liberty, but to have more, so that if you engineer cells correctly, they could go to either planet and survive just as well in either way, possibly with epigenetic modification systems built in. And again, this was kind of hypothetical not that long ago. The idea of making a Earth similarity index and looking at planets that are nearby or the moon, we've built up based on ESI metrics, another version of this here, which is looking at the mass, the radius, escape velocity, the flux, density, and temperature of the planet. How close is it to Earth, with Earth being 1, and how many parsecs away are some of these candidate planets? So the ESI, the Earth Similarity Index, blue is better, purple's okay, and white and below is actually not as good. So the moon's not, it has some features that are similar, but it's not a great place that's similar to Earth for living. But we've been able to find more recently a lot of other exoplanets that are starting to get close. So what's really exciting is that by the time we get to phase 8, this plot will be much, much better. You can see the first exoplanet was discovered in 1992, the first evidence in 89, but really usually viewed as 92 as the first year was really discovered. And more of them have been found by these transiting methods. So we would have, if you think about hundreds of years of now, we'd probably have tens or hundreds of thousands of candidate planets that we could think about sending to. And so then if we go back to this ESI metric, the Earth Similarity metrics, and look at other planets that we already know of today, there's already a pretty good number. And so this is really extraordinary because 25 years ago, we really had no human genome, very limited knowledge of all the human genes. Now we have routine human genomes and exquisite knowledge of most of them. And we had 25 years ago almost no evidence of exoplanets. Now we have thousands of them. And this metric, this ESI metric, what's extraordinary about it is if you look at everything that we've seen within 20 parsecs, these are all these planets that we know of today that we could potentially go to that are blue, that actually will be pretty close to Earth, that we can maybe go survive on as we speak today. And if you look at what we found, where we've looked for these planets, just in the past, you can see this is the burst of where are we finding these exoplanets. And, some are the white ones, so they're not good candidates yet. But the blue ones are in all sorts of directions. In this here, you see there's this big burst of possible exoplanets with the ESI that's at least close-ish to Earth. They're all in this one line because this is where we've been, essentially sending our scopes to look out. We haven't looked everywhere. We haven't looked at greatest density. We've only looked in this one part of the galaxy so far. But even just in that small part, we have these thousands of possible exoplanets that we could go to, which means, again, 20 years ago, we could really just 2 or 3 we could think of that were really not at all remotely suitable, whereas now we have hundreds that are actually these kind of bluer planets that are in the habitable zone. And so what that means is by the time we get towards the year 2400, 2500, it's possible we could do this idea of a generation ship. And so this is actually an old idea, first published by Van Bruin over 100 years ago, is that if something is far enough away with current propulsion methods, you'd have to have multiple generations live and die on the same spacecraft. If we find fusion reactors or we get something done faster before then, great. But if we presume no advances in propulsion technology or genetics beyond what we know today, you could have humans live and die on the same spacecraft on their way towards another planet. essentially this would be an imagining of like, well, I am a human. That sounds, but how could you do that? I think after the pandemic, living and staying in one place for a long time has gotten more palatable, but it's also revealed, how, it's also not ideal. Say, well, I don't want to be stuck in one place. But, this generation ship, if we make it big enough and include the totality of all of human culture, this actually could be something where, even though if it takes 20 generations to get to us one planet, you could make it so you could watch endless Netflix, or you could have sports. You'd have your own culture on that ship. It would really become its own ecosystem on its way to another planet, and then hopefully help humans get to another planet. And so the vision is, the hope is that this would actually occur. So let's think at what's happened across all these different technologies over the past 50 years and in this next 100 years. You can see this is kind of a summary we put in the book of what are all the different ways that you could actually map out what's going to happen. And then when we get towards the year 2500, the hope is that we'd launch the first generation ship and start to send people also to Titan. So I think eventually we'd get enough lessons to maybe have people visit Titan, and then maybe have stable systems around both Earth and Mars by that point. And by 2500, we send people on a generation ship to actually go towards a new star that would have a potentially habitable exoplanet. And so you might be thinking, why do all this? What's the point of all this? And some of it's just relatively simple, is that if you look long enough ahead, we can't stay here forever. And also we know that the Earth as its ecosystem as it stands is not permanent. So we know, for example, that 3 billion birds have vanished since 1970. It's about 1 out of every 4 birds in North America has gone away. And this is not the first time. So if you look over the past 500 million years, Earth's temperature, generally when it has big fluctuations, has led to large-scale extinction. So you can see what percentage of species are likely to have been devastated or extinct based on these different wild, large-scale fluctuations of worlds with and without polar ice caps. You can see both here. And today, as most of you know, climate change is pushing this to go up faster than it has. But in the context of history, it means that we want to try to avoid a 6th extinction if we can. And if you think about also, even if we avoid that, though, which I hope that we do, if you look in the really long term and kind of zoom out on this, actually, this 500 million year chunk is really just a blip in the time when there's been liquid water on Earth. And so once the Earth cooled about 3.5 billion years ago, we've had liquid water, which has been great. We're all swimming in this pool. But really, this is a nice but only temporary temperature canyon for Earth. And so what this means is we can't stay forever. I know it's a long time away, but I can tell you, when I was finishing the last chapter of the book and I was thinking about, well, I always thought we had 5 billion years till the Earth becomes engulfed by the sun and it's really going to be impossible to live on. But really, in about 1 billion years, the Earth's luminescence will start to increase so much that the oceans will begin to boil. And I actually got really sad. I came down and talked to my wife, and she said, "What's wrong?" I just said, I thought we had more time. And she said, "Well, but it's still a long ways away. It's 1 billion years." And I said, "Yeah, I know it is, but I thought we had, the future of humanity to look at maybe as long as 100 years in that time scale, like 5 billion years. If it's proportional, it's like saying I thought my son would live to be 100, but now you tell me he's only going to be 20 years old and die." And so it means, we have a fair amount of time, but it's not infinite time. And so if there's anything that you value in life whatsoever, to preserve it, it has to eventually go to another planet. And it's not plan B, it's just plan A. And so the other thing, so that's kind of just the tactical argument of why we should eventually go. But there's a more important one. I think it's a moral argument. So in particular, whenever you think of food chains, historically, it's always been thought of, there's kind of 3 main parts. There's producers, there's consumers, and there's decomposers. And this is for the history of life's understanding. It's kind of been these 3 components. But I would argue that essentially what we have now as humans is we represent a 4th kind of organism. We're kind of essentially the guardians. We are the only ones that are aware of this capacity for large-scale extinction, and the only ones that can prevent it as far as we know. We can also cause it, which I don't have a great track record on that, but we're also the only really humans are extinction aware. Only we can actually prevent this from happening. We're basically a 4th and unique kind of organism, which doesn't even have to be human. Eventually, maybe AI could become extinction aware and serve as guardians. I hope that they do if they become self-aware. And so what this means is, we can begin to prevent extinction or serve as these guardians. So for example, one project called Revive and Store actually had cells that were cloned, from a black ferret and keeping this alive. This is Ben Novak here has been doing great work on that, and also cloning the woolly mammoth to bring it back from extinction. And so what I think this means is that just ethically, we're the only creatures who know this. This gives us a duty to protect, really, and serve as guardians, not just for our own species, which can be a bit selfish, but for really all of organisms that we find, because life, as far as we know in the universe, is extremely rare. It's only found here so far. So it gives us a duty to protect all life, actually past, present, and future. And if you look far enough ahead, you could imagine, like, well, what if we keep doing this? We just go to one star, then to the next star. Do we eventually try and prevent the, say, implosion or destruction of the universe, or the heat death of the universe? Do we try and prevent this from happening? And I think here the answer too might be yes, because we don't know if life will come up again in, say, the next universe, that we may have some ability, and I think an ethical duty, to survive as long as we can as the only sort of guardians and safekeepers of life, of all life. And so to that end, I think and hope that everyone can contribute as much as possible to it and think more about these ideas in the years ahead. So I want to thank everyone on the Twins PI teams. It was really a phenomenal crew to work with for this mission. Thank, of course, a lot of the astronauts, particularly Scott Kelly, who gave really blood, sweat, and tears for this project. Thank, of course, members of the lab who make all this work possible and are endlessly inspiring, and also take really funny pictures sometimes. And of course, funding from the NIH, from NASA, from World Quant, the Gates Foundation. Thanks, of course, to the many large panoply of collaborators, colleagues, and friends. And of course, finally, thank you to having me here today. And I'm happy to take any questions. Thank you so much.
Awesome. Yeah, thank you so much, Chris. This idea of how we go about kind of writing our own genetic destiny is really exciting. And we have a lot of really great questions from the audience. So I want to start with a question from Boone, who asks, the gene editing required to make humans and other life forms at home outside of Earth could easily be abused over the next 500 years. How do we navigate the path safely and justly?
Great question. So there it is. I proposed a lot of multi-generational clinical trials. Most clinical trials now are over the course of a year or a handful of years, but rarely are they done to look at long-term changes to the genome or to the environment. And so I think you'd want to make sure they're safe is the first goal. The other thing is the abuse of what if someone finds some way to make someone a better criminal, or say, I have a whole description in the book about changing structures of the eye so you could see different wavelengths of light. What if someone did that so they could steal better at night or something like that? So like with any technology, it'll be a double-edged sword, so that it could be used for probably good purposes as well as potentially malevolent ones. And it would just require, being able to track it, make sure it's being, when tools are ordered to do the genome editing, there's some ability to track what it is. The way we do it today is, if you try to order synthetic DNA, we look at the sequence. Are you trying to order anthrax, for example? And if it is, it gets flagged and checked. And so you want to make sure people aren't using it for, say, bioterrorism purposes. We would probably have to do something like that with the deployment of some of the therapeutic and engineering, genome engineering tools.
Yeah, that makes sense. And sort of in that similar vein, we have a question from Nick, who asks, would the proposed edits to humans be exclusively in the somatic line, or are there proposals including germline manipulation, which would lead to heritable changes?
It's a great question. I discuss a lot. I think we have to consider both. I think if we do it correctly, the safest and like the least generational disruption would be to just do somatic editing, which is now $200 million at the NIH doing somatic editing therapies, including CAR T trials like I just described, and that we've published on a trial here actually at Cornell. I think that's the safest way, but I think there may be instances where instead of having to continue to do somatic therapeutics, for example, if an astronaut's going to a faraway planet, you have kind of two options. Do we have a really safe way to add genetic catalogs to their genome so they could survive better? If it has to be done germline because for some reason it works better, you may have to consider that. But if it could be done in a somatic fashion where you do just temporarily for that one person, that is the least risky, but you may be resource limited on a new planet. So you may not have the luxury of somatic therapies. You may say, we'll have to send people with everything we can that's as automated as possible. For example, like making your own amino acids or vitamin C, we would maybe do that natively. But that does presume that we have completely safe genome editing tools, which we don't have today. So that's why I put it about 40 years in the future that we'd start doing. There's versions called prime editing, which have many less off-target effects, but we are not yet at the perfect genome editing or writing or synthesis phase. But I think we're getting closer every year.
Yeah, and I actually had a question that's sort of related to that. So you mentioned in your book basically that we should learn from nature about some of these different genetic techniques, CRISPR sort of being borrowed from the way that bacteria protects against viruses. Do you see any kind of limitation in that realm, especially given that the environments that we would need to survive in are harsher than there are environments here on Earth?
Yeah, I actually think so. There's a chance that even all the lessons of Earth's history and evolution on this planet might not be enough for something that's a really weird, different planet. And as a later part of the book, I describe, sort of this point-to-point biology where you would learn about maybe if we have new organisms that we find on another planet, which would be cool and great, we could learn about them and then maybe even sequence them and then send the genetic code back to Earth for further study. So you don't have to ship the creature back, just ship the genetic code back. And then vice versa. So eventually, whenever we go to some other planet, including Mars, which we already probably gotten a little bit of DNA there, things will evolve there quickly, and we could then study them there, sequence it, and send it back to Earth to learn about it, and then propose modifications and send them back to the other planet. So you get this sort of interplanetary directed evolution, as they call it. Basically, you're just learning from other planets. And I think it's a great question, because we may, if we do find life on another planet, it might be so peculiar, it may not have remotely similar nucleotides that we use here. It might have this really strange adaptation, but we may need that to survive there. Yeah.
Right. That makes sense. And so we have another question from Andrea, who asks, should humans be the only things that we send to other planets, or should we also send animals?
Great. Well, we know that cats and dogs can make their own vitamin C, so they're okay there. But I think also for companionship, and just, I mean, we can't neglect the need for culture, for the cognitive and social, psychosocial aspect of human development, human just sanity. And pets are a big part of that. In a pinch, I guess you can eat them as well, I guess, if you're on another planet, but hopefully not. But I think, yeah, so actually for pets, they'd be good, but also I have a whole section in the book. It's a really fun thought experiment of, it was fun to write this part of the book, is, which creatures could we think of today would have the best likelihood of becoming sort of sentient creatures and becoming, say, extinction aware. And so I put in there, like, is it dog? Is it other primates? Is it octopuses? Those octopuses are pretty smart. So maybe we send to a watery planet like a whole battalion of octopuses that hang out, and then maybe in 100 million years we go back and they're doing their own thing. So it's a fun thing to think about. Look around all creatures on Earth. Which ones do we think would we place a bet on would eventually become, essentially planetary buddies? They could also become our enemies and maybe attack us. There's always that risk that it's not necessarily a rosy future. But I described for watery planets that most of the water-based creatures would be good candidates, like dolphins, octopuses.
Yeah. And actually, I had a question that's in that vein of not necessarily a rosy future. So I'm wondering. I guess there's also a lot, especially in science fiction, these sort of dystopian plot lines where humans don't necessarily continue to progress technologically, and we get to the point where either there's an event, or there's even a degree of information warfare to the point where humans are kind of influencing the fundamental understandings that we are getting. Does this play in at all into your thoughts about this?
It could be. I mean, yeah, I think writ large, most of the futuristic predictions are dystopian rather than utopian, it seems. So I think maybe they sell better. That could be part of it. People want to be a little bit scared when they see what's coming. I'm distinctly on the optimistic side, but there's plenty of really great books. Neil Stephenson's one recently, Seven Eves, was about essentially social dystopia and social contagion, where there were all these people trying to survive in space, but essentially they had worked themselves up through bad information and started attacking each other. And you do have to worry, especially when people are desperate or on the edge of survival, that they would, of course, become a bit less stable and maybe become, just more angry or desperate, or even just become less stable as a society. And that's even without, say, information technology being used against people and purposely deceiving people. Or that what if these planets were used as like a prison system rather than an exploration system? There's ways that it could definitely go south. But I think, it's really just like any technology. I think we may have fits and starts where even atomic energy was used first for weapons and then eventually for therapeutic purposes. And I think the same thing will be true here. We probably will have, unfortunately, if history is a guide, there'll be some fits and starts. There'll be some bad actors. There will be some people who use the technology either to make money or for evil purposes, and they have to be combated and potentially stopped. But I'm hoping it's limited, and that the information warfare also doesn't contribute too much to it.
Yeah, that makes sense. We have another question from Nick, who asks, what areas of this effort can software help with most? Are there blind spots that aren't being worked on as much as opposed to areas with lots of work, like sequences databases? And I think he was specifically talking about kind of the genomic editing area.
Yeah, there's a lot on. I described some of our sequence database. There's also a Google Genomics group. I don't know if any of them are on today, but I know they made their own genome browser for a while, which was pretty cool. So I know there's also a lot. I know actually a good amount of people use the Google Cloud Compute platform for precision medicine, for genome sequencing. So I think cloud-based compute has already really been helpful for almost every aspect of medicine, including de novo assembly and genome prediction and genome mapping and designing therapy. So I think that's in good shape. One of the hardest things is actually, I think, on the modeling of the interplay between genes. For example, the polygenic risk scores. How do we just define, if you were to build a genome from scratch, what would you put in different locations? To really mine that data, you need access to all of the molecular data and clinical metadata for ideally millions of people. So it's building out a lot of classifying algorithms. Most of them are various machine learning, support vector machine, random forest tools that I think people are probably familiar with, but then applying them to medical data sets would be very helpful, because as I've shown you right now, our polygenic risk score predictions are pretty meh, you know, they're not that great. So getting more tools to deploy, which are just large data frames that I'm sure most people here are probably familiar with. So I think it would be useful there. It'd be useful just for also, I think, you know, designing genetic circuits. There's another thing that requires a lot of programming, interface, and modeling, which also involves molecular dynamic simulation. So what are called MD simulations of just how the proteins interact with each other and then interact with drugs. But that's another area where the programming could always be helped by smart people.
Yeah. So in kind of the early phase of your book, you're talking about the genetic discovery and learning these genetic techniques. How do you see the pandemic impacting that phase? Do you think it's going to kind of slow things down or speed things up or not have any impact?
It's actually been great. Well, weirdly, I mean, the pandemic's been awful for humanity, but great for hanging out with my daughter more at home, and for you know, really getting a lot more funding towards the pure discovery efforts, particularly for pathogens. So most of it is, I want to go find pathogens that might be zoonotic reservoirs, like looking in bats, looking in other sort of people looking in pangolins, looking in any places where viruses might hide out. Also including, you know, marmosets, all sorts of animals. So the ability to deploy more just raw dollars towards that effort is really gone up almost tenfold. We just had an announcement from the CDC last week that there's going to be, you know, 1.7 billion just towards tracking the virus. So we can see evolution of the viruses happening in real time. We can find new viruses. So that's been extraordinary, and I think will be. I just hope some of it continues, because there's probably, you know, 1 trillion extant species on Earth, and we've found really tens of thousands of them with good references. So we have a long ways to go.
Yeah, that's great. And we're just about out of time. Chris, I want to thank you again for joining us. It's been an absolute pleasure having you with us today. And Dr. Mason's book, The Next 500 Years, is available now wherever books are sold. For everyone who joined us today, we look forward to seeing you at our next Toxic Google event. Please stay safe and take care.
Thank you so much. Pleasure.